Hi students, next topic is formation and detoxification of ammonia. Before going to the topic, please subscribe my channel, press the bell icon for notifications, like, share among your friends and comment. Ammonia is synthesized in the body as a result of the degradation of amino acid. That means metabolism of amino acid lead to the formation of ammonia. Synthesis of ammonia take place as a result of two processes. One is called deamination and the other is transamination. Ammonia is a toxic weak basic compound that needs to be detoxified and eliminated from the body. Ammonia derived from the metabolism of amino acids and especially that of gluconeogenesis transversion of amino acid into glucose. Glucose can be synthesized from amino acid carbon skeleton after the removal of its amino group. This removal of the amino group occurs through two similar processes, transamination and deamination. We can see what is deamination and transamination in detail. Transamination is a process of transferring amino groups from one molecule to another. There is no formation and no excretion of ammonia, thus no net change in the nitrogen amount of body. It is a process involved in amino acids in which the amino group is transferred from the amino acid to a certain alpha keto acid with the consequent formation of a second alpha keto acid and amino acid. The reaction is catalyzed by the enzyme aminotransferase, which requires pyridoxal phosphate as a prosthetic group. All transaminases contain this prosthetic group which derives from pyridoxine, a water-soluble vitamin also known as vitamin B6. The amino group from amino acids is temporarily uptaken by the pyridoxal phosphate as pyridoxamine phosphate prior to its donation to an alpha keto acid. All amino acids except lysine, threonine, proline and hydroxyproline participate in transamination process. Here shows the transamination of amino acid. See one alpha amino acid, see glutamate, alpha ketoglutarate to alpha keto acid and alpha amino acid become glutamate. See like this. Role of pyridoxal phosphate in transamination is also shown. Deamination is a process of removing amino groups from one molecule in order to reduce the amount of nitrogen of the body through ammonia synthesis and elimination. It is a process occurring in the liver during the metabolism of amino acids. The amino group is removed from the amino acid and converted to ammonia, whose toxic activity is cancelled by the conversion into urea, which is eventually excreted. The glutamate dehydrogenase GDH enzyme present in liver occupies a central role in nitrogen metabolism. Glutamate amino acid is cleaved into alpha ketoglutarate and ammonia, a reaction catalyzed by GDH in a process called deamination. Glutamate is the only amino acid that undergo oxidative deamination at a relatively high rate. The formation of ammonia from the amino group thus occurs mainly via the amino group of glutamate. See here, as a result of deamination, amino group is removed, ammonia form and convert into urea. Here from the carbon skeleton, glucose, acetyl-CoA semi and ketone bodies are synthesized. So oxidative deamination is shown in this figure. So far we discussed about deamination and transamination process and synthesis of ammonia. Next is, what is the difference between deamination and transamination? In transamination, transfer of an amino group from one molecule to another, especially from an amino acid to a keto acid, take place. In deamination, removal of an amino group from an amino acid or other compounds. In transamination involves synthesis of non-essential amino acid, deamination involved in the breakdown of excess protein. Transamination occurs in all cells of the body. Deamination occur only in the liver. Transaminases or aminotransferase catalyze transamination reaction. Deaminases catalyze deamination. Transamination results in an exchange of amine group with a keto group. Deamination results in the elimination of ammonia. Glutamic acid is the main form of amino acid produced in transamination reactions. In deamination, glutamic acid is the primary form of amino acid which undergo deamination. Transamination is reversible, deamination is irreversible. Ammonia is highly toxic. So how the detoxification of ammonia take place? Ammonia is rapidly removed from the circulation in the liver, converted to a water-soluble compound known as urea. Ammonia is toxic to the central nervous system because it reacts with the alpha-ketoglutarate to form glutamate. As a consequence, the depleted level of alpha-ketoglutarate impart the function of the citric acid cycle in neurons, depriving them energy production. Furthermore, glutamate is a potent CNS neurotransmitter. Thus, any significant increase in the concentration of glutamate could have abnormal effect in synaptic transmission. Here we can see the elimination of ammonia from the body. You know, ammonia is formed as a result of the degradation of amino acid. Then ammonia is highly toxic and that may damage the brain. So when ammonia is formed, it should convert into a non-toxic substance called urea, and elimination of ammonia take place with the help of urea cycle. Ammonia can be produced by the breakdown of amino acid or by gut bacteria in human. If the level of ammonia in the blood become too high, then it become toxic to the brain. The urea cycle removes ammonia from the blood and makes urea, which is eventually excreted as urine. Dear friends, thank you for watching. Don't forget to leave a like and please subscribe my channel.